Welcome to the Deep Dive. Today, we're diving into a specific piece of research, an 8-page paper from 2025 published in the International Journal of Molecular Sciences.
That's right. It's a really concise yet thorough review looking at neuromuscular disorders linked to the HSPB8 gene.
And our aim here is to, really walk through this paper's key findings for our listeners who are mostly medical professionals.
Exactly. It synthesizes quite a lot of information on HSPB8's role and the diseases tied to its mutations. It's a useful update, being from this year, 2025.
Okay, let's start with the basics then. HSPB8 itself, what's its fundamental role, according to this paper?
So HSPB8 is a small heat shock protein. It's crucial for protein quality control in our cells. Think of it like a cellular chaperone.
A chaperone, meaning it helps other proteins.
Precisely. It recognizes damaged or misfolded proteins and stops them from clumping together, from aggregating, which can be toxic.
And it does more than just prevent clumping.
Yes, the paper stresses its role in something called CASA, chaperone-assisted selective autophagy. Basically, it helps tag these bad proteins for disposal.
Okay, say. And why is HSPB8 particularly important for neuromuscular health?
Well, it's highly expressed in skeletal muscle, in the heart, and also in neurons. Tissues that are obviously critical for movement and nerve function.
Makes sense. So this 2025 review divides the HSPB8-related disorders into 3 main groups. Let's take the first one, distal hereditary motor neuropathy, or dHMN.
Right, dHMN. The paper explains this involves degeneration specifically of motor axons. So you get weakness and muscle wasting, mostly distally hands and feet, but without sensory loss.
No sensory loss, that's a key point. And the genetics.
It's genetically diverse, dHMN overall, but the HSPB8-related form is typically autosomal dominant. The paper references the initial link back in 2004.
Ah, yes, with those specific mutations found in European families, the lysine 141 ones.
Exactly. PLS141A is and PLS141 GLU. That was the first time HSPB8 was flagged as a cause for dHMN.
And what's a typical clinical picture described in this 2025 review? How does it usually present?
Onset is often mid-teens to mid-20s. It usually starts in the lower limbs. Weakness in the big toe extensors is very characteristic.
Was the big toe initially?
Often, yes. Then it progresses, sometimes leading to paralysis in those distal muscles within about 10 years, and eventually it can spread proximally into the upper limbs too.
Are there other symptoms mentioned?
Sometimes cramps or fasciculations, but crucially, usually no sensory issues, though older patients might lose some vibration sense.
And how common is this HSPB8 form of dHMN?
The paper cites studies suggesting it's one of the less common types of dHMN, actually.
Okay. What about diagnostic clues? Anything specific in blood work or scans?
Serology isn't usually very helpful, maybe slightly elevated CK in some rare cases. Electrophysiology, like EMG and nerve conduction studies, shows those distal symmetric chronic neurogenic changes.
And imaging? Brain, spinal cord?
Generally normal, according to the review. Nerve biopsies show minimal changes, maybe some fatty replacement in muscle, but nothing too dramatic. Very few onion bulbs, for example.
Right. So moving to the second category of the paper, Charcot-Marie-Tooth disease type 2L, CMT2L. How does this relate?
Well, CMT is another inherited peripheral neuropathy, quite common actually, but CMT2L is a specific axonal subtype.
And the HSPB8 connection.
This is interesting. The paper points out that in 2004, the same mutation linked to dHMN PLS141-AMBOR was also found to cause CMT2L in a large Chinese family.
The same mutation. Wow. Okay, so how does the clinical presentation differ then for HSPB8 CMT2L?
The onset age is similar, maybe slightly later, mid-teens to early 30s. It's also progressive distal weakness, mainly lower limbs, sometimes proximal too.
But the key difference from dHMN?
Sensory loss. CMT2L involves sensory nerves, so patients have sensory loss, plus often reduced or absent reflexes, hypo or reflexia. Pescovis, that high-arched foot, is common too.
And this subtype, HSPB8 CMT2L, is it common?
No, the review states it's also considered rare.
Diagnostics for CMT2L. What does the paper highlight?
Electrophysiology shows reduced signal amplitudes, CMFs, and SNAPs, even if velocities are normal. EMG shows neurogenic changes. Muscle MRI can show atrophy and fatty replacement, particularly in the legs, sometimes the thigh muscles too.
And nerve pathology. More revealing than in dHMN.
Yes, a bit more. Axonal degeneration, loss of large myelinated fibers, some thinly myelinated clusters, and even ultrastructural things like vacuoles and axons and Schwann cell issues.
Okay, that brings us to the third category, HSPB8-related myopathy, which is now called MFM13, myofibrillar myopathy type 13.
Right. This is a more recently recognized connection. It's an autosomal dominant myopathy, so primarily a muscle disease, not a nerve disease, though there can be overlap.
And what defines this myopathy clinically, based on the 2025 review?
Onset is usually adolescence to mid-adulthood, sometimes even childhood. Lower limbs are often hit first, then it moves proximal and involves axially muscles the trunk.
Axial weakness, like difficulty sitting up straight.
Exactly. Camptocormia, that forward bending of the trunk, can even be an early sign. Weakness can be asymmetric, and the beaver sign might be present. Facial muscles are usually spared, though.
What about reflexes and other features?
Reflexes are variable, normal, low, or absent. Sometimes skeletal issues. Cramps and fasciculations can occur, especially if there's also some neuropathy involved, which does happen in some patients.
Any involvement beyond the skeletal muscles? Respiratory, cardiac.
Respiratory dysfunction is definitely something highlighted in the review. It can range from asymptomatic to severe failure needing ventilation, and the timing varies a lot.
And the heart, given HSPB8 is expressed there.
Interestingly, cardiac symptoms seem uncommon in this specific myopathy. Cardiomyopathy was reported in just one family mentioned in the literature reviewed.
How is HSPB8 myopathy diagnosed then? What does the paper say?
CK levels can be normal or elevated, sometimes up to around 2,000 IUL. EMG typically shows myopathic changes, short duration potentials, maybe fibrillations, but you can also see neurogenic signs mixed in.
And muscle imaging, MRI.
MRI shows signal changes progressing to fatty replacement, often patchy and asymmetric in the legs and thighs. The paper notes specific patterns of which muscles tend to be involved or spared.
Muscle biopsy must be key here. What are the pathological hallmarks described?
Very complex. You see dystrophic features, myofibrillar disorganization that's characteristic, and rimmed vacuoles, though the amount varies. Also, fiber size variation, internal nuclei, splitting, inclusions.
Protein aggregates too, I assume.
Yes, aggregates containing proteins like myotylin, desmin, P62. Crucially, the review notes that HSPB8 itself is found within these aggregates, along with other proteins like BAG3 and TDP43. Electron microscopy confirms the myofibril breakdown.
Okay, let's shift focus slightly to the gene and protein itself, based on the review's discussion, the structure of HSPB8.
It's on chromosome 12, 3 exons, makes a 196 amino acid protein. It's an ATP-independent chaperone, meaning it doesn't need ATP energy to function directly. It has that conserved alpha-crystalline domain, the ACD.
Right, flanked by N-terminal and C-terminal domains. What do?
The ACD is the core chaperone part. The N-terminus interacts with other heat shock proteins, and the C-terminus grabs onto the substrate proteins. It usually works as a dimer and interacts closely with BAG3 for that Kaiser pathway.
So functionally, the paper reinforces its role in
What exactly?
preventing aggregation, helping refold proteins with HSP70, and driving that Kaiser degradation pathway. Super important for neuromuscular health, as the paper emphasizes. It's also involved in stress responses, like stress granule dynamics.
And mutations in other related proteins, other HSPBs, also cause neuromuscular issues.
Yes, the paper mentions this, highlighting that the whole family is critical.
The review mentions a figure 3, linking mutation location to the disease type. Can you summarize that correlation?
Yeah, it's quite neat. Generally, missense mutations, single amino acid changes in the N-terminal or alpha-crystalline domains tend to cause dHMN or CMT2L.
Like the lysine 141 hotspot.
Exactly. Whereas frameshift mutations near the end in the C-terminal domain, which make the protein longer, are typically linked to the myopathy MFM13.
Interesting. And the inheritance pattern. Mostly autosomal dominant, as we said, but the paper notes, de novo, spontaneous mutations can happen too.
So what are the proposed mechanisms? How do these mutations actually cause disease, according to the review?
For the missense mutations, like at K141, it's thought they might reduce the chaperone ability or destabilize the protein, maybe a partial loss of function or altered function.
And the frameshift mutations causing the myopathy.
Those seem to be more of a toxic gain of function. The elongated protein forms these insoluble clumps, aggregates that gum up the works. They seem to trap important components of the TACEP pathway and other necessary proteins, disrupting proteostasis, protein balance, and even muscle cell development.
Okay, toxic gain of function. Does the evidence from animal and cell models discussed in the review support this?
Yes, largely. Cell models show mutated HSPB8 loses its protective functions and can impair autophagy. Knockout mice lacking HSPB8 entirely are surprisingly okay locomotor-wise, which argues against simple loss of function being the main problem in humans.
So the mutated protein itself is the issue.
It seems so. Knock-in mice with the human K141N mutation develop motor neuron and muscle problems, including protein aggregates, really mimicking the human disease. This strongly points to that toxic gain of function. Even heterozygous mice show changes.
Fruit fly models, too.
Yes. Drosophila studies also show different effects depending on the specific mutation, reinforcing the idea that the type of mutation matters. So overall, models support this toxic gain of function idea involving messed up chaperone activity and autophagy.
Given this understanding, what does the 2025 review say about potential therapeutic strategies? What's being explored?
Well, one idea is to try and boost the remaining functional HSPB8 or induce its expression. Enhancing autophagy generally to help clear the aggregates is another major avenue.
Are there specific compounds mentioned?
The paper lists a few small molecules identified as potential HSPB8 inducers, things like trehalose, colchicine, and others. Research is ongoing there.
What about trying to reduce the toxic protein?
Interestingly, the review mentions an RNA interference study in mice targeting HSPB8 didn't really help, which again supports the gain-of-function idea. Just removing it might not be enough or could even be detrimental.
So what else looks promising?
Delivering functional HSPB8 from the outside seems promising. Using viral vectors or maybe extracellular vesicles, tiny packages released by cells to get healthy HSPB8 into affected cells. There's an example cited using vesicles from oligodendrocytes.
So several different angles being pursued. The review also points out some limitations in the current research, right?
Yes, definitely. The rarity of these diseases is a big one. It makes it hard to fully capture the whole clinical picture or run large studies.
Well,
The possibility that HSPB8 mutations affect other organs needs more study. Also, current models, while useful, don't perfectly replicate human complexity, genetic background, environment, all that plays a role.
so we might only be seeing part of the picture.
Exactly. And understanding why the weakness and degeneration happen is still ongoing. Plus, testing therapies is tough due to the rarity and variability between patients.
Okay, so wrapping up based on this 2025 review's conclusions.
Yeah.
What are the main takeaways?
The key message is the expanding range of disorders tied to HSPB8 motor axons, nerves, muscles. The myopathy phenotype is variable, and respiratory monitoring is important. Cardiac issues are less common, but worth keeping in mind.
And the genotype-phenotype link.
It holds generally. Missense for neuropathy, CTD frameshift for myopathy, often involving issues with proteostasis and sarcomere structure in the muscle.
And treatment.
Still no cure or way to reverse deficits currently, but as we discussed, several therapeutic strategies are actively being investigated.
So this 2025 review really provides a solid current overview. It highlights how crucial HSPB8 is for neuromuscular health and how different tweaks to this one protein can lead to distinct diseases.
Absolutely. That connection between the specific genetic change and the clinical outcome, the genotype-phenotype correlation, is really striking. And the toxic gain-of-function idea seems central for many of these mutations.
The research into therapies, from boosting HSPB8 to delivering it, sounds hopeful, even if it's early days.
It is. The paper does a good job of laying out the current state of knowledge and the directions research is heading.
Which leads to a final thought, perhaps, for our listeners. We've seen how critical protein quality control, involving HSPB8 and casafat, is in these specific disorders, as detailed in this 2025 paper. So how might a deeper dive into this specific pathway, the HSPB8 SASA system, potentially unlock broader therapeutic ideas for other neurodegenerative diseases that also feature protein misfolding, things beyond these particular neuromuscular conditions?
That's a great question. It definitely suggests that understanding these fundamental cellular housekeeping mechanisms could have really far-reaching implications. A lot to think about.
Indeed. Well, thank you for walking us through this insightful 2025 review today.
My pleasure. It was a good discussion.